So this lesson is the first lesson that I directly am going to give you guys on the reproductive system. Usually, as I said, this content would come after Professor Fink's discussion, but I will catch you up to speed and then we will continue, and then you'll go back and watch that content later. Essentially, the reproductive system of course, contains organs with two different primary functions. Of course, there are many other functions which he will get into in the Ed puzzle. But for now, the primary functions of the reproductive system are essentially to create gametes. All right, so when I say gamete, I mean essentially sperm or egg. What makes these cells different from normal cells is that they have one copy of DNA instead of two copies of DNA like the rest of our cells have. And the idea behind this is that the male gamete brings half of the DNA, the female gamete brings the other half of the DNA, and when they join together, they form a genetically unique individual. So they have half of mom's DNA, half of dad's DNA, and they can create a new individual. Right. Good stuff, guys. The other primary function of the reproductive system is to create hormones. So it is a very powerful endocrine or series of endocrine organs. So sex steroids include testosterone, progesterone, estrogen, a series of androgens. So a lot of different hormones that, of course, modify the activity of the entire body. They can drive the development of secondary sex characteristics like growing a beard, like growing breasts. They can drive behavior. They can drive cycles, as we're going to see here today. Again, the reproductive system is creating gametes and it is creating hormones. And that is really going to be the focus of today's lesson here. Ultimately, there are a lot of different reproductive organs, and we're going to go over, I'm going to go over the female side of things today, and Professor Fink will talk to you about the male side of things in the Ed puzzle. But ultimately, it is the gonads, it is the testis, right? Not testicle, the testis, which is right here. The testis produces both gametes and hormones. Again, you're going to learn in more detail about this later, but essentially the testis actually is composed of a series of tubules. These tubules are called seminiferous tubules, and within them, and kind of around the perimeter here, is where both testosterone and sperm are created. And so we're going to start a conversation today with looking inside of a seminiferous tubule. Ultimately, sperm are made within the border of the seminiferous tubule. They mature and are passed through the seminiferous tubule, up the tubules, and they are stored in what is called the epididymis, which is what this is right here. And ultimately, during an ejaculation, the sperm are going to be propelled via peristalsis, right? So they're not swimming while they're in the man's organs. Instead, they don't actually start swimming until post-ejaculation. So ultimately, this tubule, right, which is continuous at the seminiferous tubule, becomes the vas deferens or ductus deferens, and it is going to propel sperm up over the pubic bone, over the urinary bladder, behind the urinary bladder, joined together with contents from the seminal glands or seminal vesicles, joined together with contents of the prostate gland, ultimately before being ejaculated. So again, you'll learn all about that kind of stuff in the other presentation. I just wanted you to have kind of a big picture of what is going on here. Again, we're starting our discussion today with looking at what is happening on the periphery of these seminiferous tubules. Okay. So the creation of gametes, right, in this case sperm, involves the process of meiosis. And so you may or may not remember this from A&P1 or from an introductory biology course, but essentially meiosis is the process of taking one cell and making 4 daughter cells out of it, or 4 sperm cells. So the actual cell division process is called meiosis, and the process of making fully mature sperm that are capable of swimming, that are capable of penetrating the many layers on the surface of the egg, is called spermatogenesis. So genesis, generation of sperm. Okay. This entire process is going to begin with a specialized type of cells called a spermatogonium for singular, or spermatogonia plural. These cells are genetically identical to every other cell within the man's body, and they essentially provide a population of stem cells from which sperm are going to be derived. So before spermatogenesis, the creation of sperm, can begin at all, these germ cells, right? So these stem cells have to complete mitosis, right? So mitosis, as opposed to meiosis, and again, maybe this is a review, maybe this isn't, but mitosis is the process of essentially copying an existing cell. So the spermatogonium has 2 copies of DNA, just like every other type of cell within the man's body. It makes a copy of itself, right? Given the process of mitosis, and all of those little details are not necessary for us to go into, for you to memorize and spit back at me on an exam. Just mitosis, you're making a copy of a cell. Start with a diploid, or 2 copies of DNA cell, and with 2 of these cells. Okay. Ultimately, one of those daughter cells, right, given the process of mitosis, is going to stay behind and is going to remain a spermatogonium. Right? It's going to stay a stem cell. So maybe you remember from AMP1 talking about the stratum basal, right, the deepest layer of your epidermis, or the outer layer of your skin. And so those cells in the stratum basal are constantly undergoing mitosis. They are dividing and dividing and dividing. And one of those cells is going to move up through your skin, right, and actually become your skin cells. And the other one is going to remain behind in the stratum basal and stay a stem cell, right? Its job is to make more skin cells. Here, the spermatogonium, right, their job is to make cells that are ultimately going to complete meiosis, right? They're going to become sperm cells. Okay. So this is a very simplified diagram of meiosis 1 and meiosis 2. Again, meiosis starts with one of these daughter spermatogonium cells, and it ends with 4 genetically unique haploid cells. So 1 copy of DNA versus diploid, 2 copies of DNA. And so in addition to creating 4 haploid cells, the process of meiosis 1 and meiosis 2 is also completing the process of genetic recombination. And this isn't a term that you will need to be able to spit back at me, but I want you to see that your chromosomes themselves, right? Your physical chromosomes, 1 copy is from mom, 1 copy is from dad. Each one of those chromosomes wasn't passed down directly from your grandparents. Instead, during your parents' process of spermatogenesis and oogenesis, those chromosomes actually mixed up a little bit. So there's a process called crossing over, or synapsis, and essentially those chromosomes are going to mix together, and some of grandma's genes and grandpa's genes are going to switch around. And so the chromosomes are actually unique. They're not exactly like your parents. They are a little bit different. Okay. Also, just completely by chance, the order in which these chromosomes actually line up before the cells divide is going to create, again, unique cells down here, right? So one of these cells is not at all identical up here. It has its own unique combination of grandma's genes and grandpa's genes. Okay, so there are a lot of ways in these processes, on top of who you choose to mate with and all that kind of stuff, that assures that your offspring are different than you. And so this makes a lot of sense. We want our offspring to hopefully be better off than we ourselves are. And so we kind of mix up the genes in hopes that some combination is the magic combination for whatever that kid is going to experience for the rest of their life. So take home message here, a stem cell divides via mitosis. One cell stays behind, is going to make more stem cells. The other daughter cell is going to begin the process of meiosis 1 and then meiosis 2. There is genetic stuff happening to create 4 genetically unique haploid, 1 copy of DNA cells, which are going to grow up to be sperm. Okay. And so this process produced the gametes. The sperm itself is actually short for a longer term spermatozoa. And so the zoa here should indicate zoo or something that's animal-like. And so this technical term for sperm is just referring to the fact that spermatozoans have a flagellum. They swim because of the flagellated movements of the cells. And so a little bit more terms here. See, my window here is in the way. I'm going to be showing you a lot of different images here from a textbook. All of them at the top show the edge of the seminiferous tubule. Okay, so this here, think about how a tubule is round. And so this here is kind of like a little pie wedge out of the side of the seminiferous tubule. Yeah, so it starts with the spermatogonium. Those are the stem cells we need, or males need to continually make more of these stem cells because, again, one of the daughter cells is going to produce 4 of these spermatozoans. So we need to always maintain a population of these stem cells. Okay, so the spermatogonium is the 2N. 2N means 2N copies of the DNA. Okay, so again, this starts on the edge of the seminiferous tubule and is going to proceed down towards the lumen of that seminiferous tubule. Here we can see mitosis producing 2 genetically identical diploid cells. Okay. One stays behind. It is still called a spermatogonium. The other cell is actually going to physically be pushed deeper and deeper into the seminiferous tubule. This is called a primary spermatocyte. Okay, so primary spermatocyte, remember, cyte means cell, and then spermato, this is going to grow up and become a sperm. The primary spermatocyte is going to begin the process of meiosis 1, producing 2 secondary spermatocytes. Note that these spermatocytes are still kind of sort of together, so they haven't actually completed the process of cytokinesis or the full division and separation of the cytoplasm. Instead, they kind of hang out together and they move down to the lumen together. These secondary spermatocytes are going to divide once again. Again, we have all of this like mixing up of the DNA and rearranging themselves on the metaphase plate and lots of different things that is essentially assuring that each individual sperm is completely unique. Okay. So each one of these secondary spermatocytes is going to divide into 2 more cells. Again, they're not completely separated, but they are individual cells. Yes, so the spermatogonium is diploid, thus the 2N here, so 2 copies of the DNA, right? So for this man, 1 copy would have been from his mom and the other copy would have been from his dad. Down here, we mix up mom and dad's DNA so that the sperm themselves are unique. I understand a lot of kind of genetic stuff, a lot of cellular stuff going on here. Okay. So each secondary spermatocyte is going to divide into 2 spermatids. So when this is, when this is happening here, 1 cell divides into 2. They're still connected together, but now we have these spermatids that are fully haploid. And so haploid again means 1 copy of DNA. The DNA is unique from either of this man's individual chromosomes. So these spermatids are non-motile. So essentially they cannot swim yet. They do not have a flagellum. They do not have a lot of other mechanics that are actually necessary to be a true sperm, but genetically they're sperm, right? So they are immature at this point. We can see that they are called early spermatids. They will continue to grow up and develop into something that looks a lot more like a sperm. The final stage of this process is called spermiogenesis. And so this is essentially taking these genetic sperm, right? So their nucleus isn't going to change at this point, but it's taking this nice little round cell here and it's growing onto it a couple other components, which we are about to talk about. No. So I know it feels weird, but the lecture and the lab are two separate classes that are kind of the same class. So all of this content is not on your lab practical. Surely it is related because you have learned about the different structures of the reproductive system, but this is purely lecture content. And so looks like lab is all done tonight. Oh my goodness, that's exciting. Okay, so these late spermatids continue to mature to become the final spermatozoans. Note in this image that there is this tiny faint little line here. That is, as we're about to see, the edge of kind of this ring making up the seminiferous tubule. So these spermatozoans are in the lumen. And as I said before, the seminiferous tubules, as well as the epididymis and the vas deferens. So all of this tubular structure throughout the male reproductive system can move the spermatozoa along via peristalsis. So kind of squeezing different sections of the tubule to milk along the sperm. They're not swimming. They're not wasting any energy moving around in the male reproductive tract. Okay. All of this process, right? So everything from way up here all the way down to this little line here, this is all actually embedded within a group of cells that is called the cystenocytes, which is the more common term now, or Sertoli cells. Right? We've seen this a lot in this class and even last semester, I'm sure. Why name something once if you can't name it 2 or 3 times? But really, we are trying to move away from this guy discovered these cells. We're going to name it after him and instead move towards terms that actually explain where it is, what it's doing, what it looks like, something like that. And so these cystenocytes are cell that sustain the process of spermatogenesis. Let's talk a little bit more about them because there are even more names and even more functions. These dividing cells are embedded within nurse cells, which are called cystenocytes or called Sertoli cells. For a little bit of a prettier diagram here, right? So here's the testis. Pulling one of these seminiferous tubules out, we can see that it is this ring structure with yellow making up the main body of the tubule. The white in the center is the lumen. Okay, so on the edge of the seminiferous tubule, this is where the process of spermatogenesis begins. And then the cells divide and divide and divide and mature and ultimately are pushed down into the lumen of the tubule. Now, the yellow is what we're focusing on right now. This is a nucleus of a big, really funny looking cell. That cell is a cystenocyte or a Sertoli cell or nurse cell, whatever you want to call them. And so what do these things do? How do they sustain this process? Well, the cystenocytes physically move the spermatocytes towards the lumen. All right, so every subsequent cell division, the cystenocytes push the cells down farther and farther closer to the lumen of the tubule. Furthermore, they're not just like holding on to them and directing their movement, but they're actually producing the chemical mediators that are going to regulate how quickly this process is going to proceed, right? How much sperm is necessary, how many chemicals are around, like what is the brain telling us to do, what is, so the brain, the hormones, etc. What rate is sperm needed. So these guys are the ones that are responsible for doing that. Specifically, and as we're going to go into more detail with in a moment here, the gonads, so testes in male, ovaries in females, are regulated by the HPG axis, the hypothalamus, anterior pituitary, and then gonad. And so the anterior pituitary gland produces a couple different hormones that regulate the gonads, specifically FSH, or follicle stimulating hormone, as we're about to see, triggers these cells, these asthenocytes, to release a chemical called androgen binding protein, or ABP. Okay. ABP essentially binds to androgens, right? Androgen is another kind of generic term for testosterone and other male hormones. And so the androgen binding protein is essentially going to hold testosterone in the testis as opposed to allowing it to circulate throughout the body. And that is directly going to trigger the creation of more sperm. Furthermore, these asthenocytes are joined together by tight junctions. Right, so if you remember, there are a couple different ways that cells can be held together. Some of them connect the cytoplasms and make, essentially multiple cells into one big unit, kind of like in the heart. Other cells essentially keep the cells from tearing apart, even though there's a lot of movement. So for example, in the digestive system, those epithelial cells are held together really tightly by different junctions to make sure that they don't tear apart and allow partially digested food into your gut. That would be super bad. Here, these tight junctions essentially separate the superficial side of the seminiferous tubule, so around the edge here, from the innermost portion, right, so the deeper portion of this tubule. And so this is really important, this compartmentalization of the seminiferous tubules. Unlike in females, which we're going to talk about tonight as well, spermatogenesis does not begin until the man reaches puberty. And so the development of the immune system occurs long before the man's testes start making these genetically unique cells. And so as far as the immune system is concerned, these are not self cells. They're not part of the man's body, right? They are purposely genetically unique. And because the immune system has never seen these cells before, right? Because, however many years until you reach puberty, you've never seen these cells before. And so when spermatogenesis begins, the immune system, if it could see these cells, would attack them and eliminate the sperm from the body. And so obviously that's not ideal. The goal here is to have offspring, and so you don't want to attack half of the equation here. So these tight junctions between the asthenocytes essentially prevent the immune system cells from getting closer to the lumen. So all of these cells, which are genetically unique, they're not technically self cells, the immune system cannot see, but it can see these cells up here. Remember that this was a stem cell. It created two genetically identical spermatogonia here. One of them is going to become sperm, the other remains a stem cell. And so these, the immune system recognizes, it is allowed to see them. These ones, it cannot. Okay, and so this concept, the structure is called the blood-testis barrier. It prevents the man's immune system from attacking his own sperm. Furthermore, the asthenocytes physically sustain the developing sperm. So this image right here is kind of upside down because it's from a peer-reviewed paper, not from a textbook. And so here is the edge. Right, here is the basal compartment, and way down here, this is the adluminal compartment. As we can see, there are some complex junctions, so some complex proteins that are more or less binding and allowing these developing cells to interact with the asthenocytes. This allows for direct communication back and forth with chemical signals as well as nutrients. right, water, etc., to actually sustain and nourish the growing sperm. So the so the cells undergoing meiosis, right? So the primary spermatocyte is undergoing meiosis to create 4 sperm. Those cells there are in the adluminal compartment, and the stem cells are in the basal compartment. Let's see, the cystinocytes have even more actions here. They are going to phagocytose faulty cells and excess cytoplasm. So first of all, if there is a mutation in one of these cells up here, or even one of these cells down here, it is the cystinocyte's job to essentially recognize that and, commit cell murder here, right? Or essentially tell the developing sperm to commit apoptosis or cell suicide. And so this prevents or reduces the chance of genetic mutations being passed along to the offspring, right? It's better to kill off a sperm or two than to risk producing an offspring with genetic mutations. Also, remember that these cells are still kind of joined together. They move down through this compartment all as one big lump. And so once the cells are good to go up the lumen, essentially it is the cystinocyte's job to trim this excess cytoplasm and liberate the sperm into the lumen. Okay. Also, the cystinocytes secrete testicular fluid. So this is not semen yet. That comes from other places in the reproductive tract, but this is testicular fluid. Essentially, you can't move the sperm unless they are suspended in fluid. And so this fluid is simply for transporting them all the way up here to the epididymis. So to look at some histology here, this is a seminiferous tubule. We can see the round tubule, and these are very densely packed. I don't know if you guys got to see in the lab a gonad dissection. If we were in person, you guys would be dissecting ovaries and testes yourselves. I'm not sure if you got to see that though, but essentially the entire testis is made up of densely packed seminiferous tubules. Here we can see those dividing cells ultimately becoming sperm way down here. And so you can actually even see some of those chromosomes are bundled up nice and neatly for the cell division process. The cystinocytes or Sertoli cells are lighter pink, and so we can see all of these cells are like suspended within. They're held and nurtured by these cystinocytes. Surrounding even the cystinocytes and all of these spermatogonia, spermatocytes, are myoid cells. And so the MYO is a root that means muscle. And so a take-home message here is that the seminiferous tubules are muscular. They are going to complete peristalsis just like we saw in the digestive tract. And one other type of cell that I haven't mentioned yet that's going to become very important within the next couple slides here are the interstitial endocrine cells, formerly called Leydig cells, but I'm sure that you'll hear both of these terms, and I kind of go back and forth between them as well because I learned them as Leydig cells as well. But essentially, these are interstitial, right? They are between the cells, they are within the tissue itself, and they are endocrine in function. And so it is the interstitial endocrine cells on the outside of the seminiferous tubules that produce the sex steroids, so testosterone, androstenedione, even things like estrogen and progesterone can also be produced by the testes. And so these are triggered to produce these sex steroids by the hormone LH, or luteinizing hormone. Right, so we're going to see here in just a moment that the anterior pituitary gland releases 2 different hormones. You need both of these hormones to make both the sex steroids as well as the sperm itself. And so the HPG axis, usually in AMP1, this is like tacked on to the very last bit of the class. And so I understand if this seems completely foreign, it really is kind of glossed over, but we're going to go into it in hopefully appropriate detail. HPG. The H is always referring to the hypothalamus. So this is part of your brain that is going to connect to your endocrine system and unite the entire body's response. The hypothalamus generally produces releasing hormones. Right? So specifically, the hormones that are produced are going to communicate directly with the anterior pituitary gland. So the P stands for anterior pituitary, not posterior, but anterior. And these hormones are called gonadotropins. And so some endocrine roots here, tropin or tropic essentially means triggering the release of something else from where? From the gonads. And so the hormones released by the anterior pituitary gland are responsible for talking to the gonads and telling them to release another type of hormone. The anterior pituitary hormones in question are LH luteinizing hormone and follicle stimulating hormone. Now, both of these hormones act in both the male and the female reproductive system, but they are both named according to their action in the female reproductive system. Okay. Ultimately, these hormones are released from our anterior pituitary, so pretty much right above the roof of our mouth is where that sits in the little Turkish saddle, the sella turcica of our skull. These hormones are released into the blood. They bathe over the entire body via the cardiovascular system, the blood vessels, and ultimately they bind only to cells that have receptors for them. In the testis in particular, so in the gonad of the male, LH is going to bind to the cell which starts with L, right? Very conveniently, those Leydig cells or interstitial endocrine cells bind to LH, and in response, the Leydig cells produce testosterone. FSH is going to bind to receptors on Sertoli cells, right? So FSH binds to the cells that starts with S. Sertoli or cystinocyte both starts with S. FSH. In response to FSH, the Sertoli cells or cystinocytes are going to produce androgen binding protein. Also, they are going to produce another hormone called inhibin. And as the name suggests, this hormone is responsible for inhibiting the action of the reproductive system. Okay, so essentially, all of these work together with negative feedback. Right? More GnRH is going to produce or lead to the production of more LH and FSH. LH and FSH lead to the production of lots of testosterone, lots of ABP, and higher levels of these things, as well as inhibin, is essentially going to put the brakes on these structures up here. And so this is a classic negative feedback HBA, HBG, HBT axis kind of situation. Here is the hypothalamus, releases a hormone that has a positive impact on the anterior pituitary. The positive impact leads to the release of these two gonadotropin hormones. These guys are going to bind to the gonads themselves. And in response, these cells are going to release testosterone. Right? Testosterone, of course, has its own impact on target cells throughout the body, leading to the development of secondary sex characteristics, leading to the development of different types of behavioral traits, etc. Okay? And the more testosterone that is produced, and certainly the more inhibin that is produced, the more of a negative feedback, negative impact is going to be exerted on the two organs farther upstream. Okay, so the idea here is that we don't want to start producing testosterone one day and then like more and more and more and more and more and more. Instead, you want just the right amount of testosterone. So if the testosterone levels get above some target threshold as determined by your own genetics, your own body, your own lifestyle, if those testosterone levels get beyond a certain threshold, then the hypothalamus and anterior pituitary are going to say, okay, we have enough. I'm going to pull up on the GnRH, LH, and FSH. We have plenty of testosterone in the body. Okay, so again, negative feedback, classic example, works the same way in the female reproductive system, except for some funniness around ovulation time. We're getting there. So essentially, this diagram has been converted into some drawings of the different organs involved. This is the hypothalamus. Right, so under your cerebrum, kind of to the front of your diencephalon. This is the pituitary gland. Remember that the pituitary has a posterior side. We're not talking about that right now. And an anterior lobe. Right, so this is the anterior pituitary gland. This here is the seminiferous tubule, like we just looked at. Yellow are those Sertoli cells or cystinocytes. Here is the edge of the seminiferous tubule. Here is the lumen. Okay. So the HPG axis and all of these different mechanisms are going to work together to regulate spermatogenesis. Again, one day you don't want to start making sperm and then make way too much. You don't want too little. You don't want too much. You want just the right amount of testosterone, just the right amount of sperm. So to initiate this process, the hypothalamus again releases GnRH. This is a portal system, so this hormone is released into a capillary bed here. The blood delivers GnRH directly to the anterior pituitary, right? Another capillary bed, talks to the anterior pituitary cells. And in response, the anterior pituitary gland releases these two hormones we just discussed. FSH, as we now know, is going to, again, circulate throughout the entire body, but only bind to the cells that have receptors for FSH. In this case, the FSH receptors are on the Sertoli cells or those cystinocytes. In response to FSH binding to the Sertoli cells, those cells are going to start creating ABP and secreting this androgen binding protein. Now, at the same time, LH is released into the blood as well, and it binds only to those interstitial endocrine cells or Leydig cells that are going around the outside of the seminiferous tubule. In response to LH, the Leydig cells are going to produce testosterone. Okay, and so a little bit about what exactly is happening in response to LH. To rewind a little bit, testosterone is one of many sex steroids. In fact, it's one of many steroid hormones. All of these hormones are ultimately derived from cholesterol. Now, as we can see, there are many different shapes and many different names of these hormones. The metabolic pathway, essentially the direction that this reaction takes, is determined by the presence of other hormones. So, for example, cholesterol, right, when a cell receives a certain message, can become progesterone, progestation. So this is the like pro-pregnancy hormone right here, right? So again, given a certain stimulus, cholesterol can become progesterone. On the other hand, cholesterol can become cortisol, right? Cortisol, the stress hormone, right? We all know and love, right? So the stress hormone is just a couple steps away from the pro-pregnancy hormone. Not going to go there. On the other hand, cholesterol, given different signals, can become aldosterone. Remember, we just talked about aldosterone. This is a hormone that is going to promote sodium retention and therefore increase blood pressure. Right? So all of these hormones are very much related. And the only way that we get one hormone and not the other is if we get just the right signal at just the right time. That's a great question. So what type of cholesterol is HDL or LDL? HDL and LDL are actually the names for proteins that transport the cholesterol around the body. LDL is actually, they're both made by the liver. Essentially, you eat cholesterol in your food. It is taken up via those lacteals to your liver, and your liver packages the cholesterol and the triglycerides and fatty acids and all of these different things into either a big, clunky, sticky LDL molecule or a little zippy HDL molecule. Okay, and so it is kind of your liver's decision, your body, a genetics decision, how much of each of these packages to make, but cholesterol is cholesterol. I know that's weird. I know we hear all the time, oh, you're good cholesterol, you're bad cholesterol. Well, what we're actually talking about are the lipoproteins, the protein packages of all of the lipids, including cholesterol. I'm not sure if that made sense, but I know it's very misleading. What you hear in like nutrition magazines and whatever, cholesterol's cholesterol. It just depends on how we package it. So they're all from cholesterol. Essentially, we can tell our cells, right, in different parts of our body, more or less what direction to go in by turning on different genes. Those genes code for enzymes. And so if we have, for example, a lot of this enzyme that we produce, then we're going to constantly be converting this molecule into this molecule. Okay. Luteinizing hormone stimulates the Leydig cells, the interstitial endocrine cells, to produce testosterone. And so essentially that is going to turn on this particular gene and convert this preliminary product, also a derivative of cholesterol, into testosterone. Okay. If we had more time, I would totally get on my soapbox about endocrine disruption and everything, but just like 2 seconds here. Maybe you guys have heard of like BPA, right? So BPA, a chemical that is a plasticizer. It is found in, or was often found in water bottles to make them squishy without breaking. So hooray, plastic, we love it, we love it. But the interaction with plastics was essentially leading to feminization of boys and of girls, like so doing some weird developmental stuff. And it turns out that BPA is essentially causing cells to turn on the gene for CYP19. So this CYP19 gene essentially codes for the aromatase enzyme. And the aromatase enzyme is the enzyme responsible for converting testosterone into estradiol, which is another term for estrogen. So even if you are a boy, you can produce lots and lots of estrogen if you have enough of this gene expressed, that is enough aromatase. Now, because of market influence, BPA has been removed from a lot of products. And so yay, you know, the power of the market, that is fantastic. It is not completely eliminated. BPA is still in the linings of your canned goods. It's still in receipts. It's still in a lot of places. And of course, we replace BPA with something. So what is that doing to us? Furthermore, BPA is by far not the strongest feminizing chemical that we interact with on a day-to-day basis. If you're interested, I have all sorts of things to talk about. But long story short, eating organic may be more expensive, but you're definitely not putting as many chemicals into your body. Yeah. So it's really hard to study in humans. So most of these studies are in animals that had the same exact pathways as these. Yeah, and BPA free. So that is really a testament to how much power we actually have as consumers. If only we do the research and know the truth, and of course stick to what we have learned, we can really change things, right? Like the fact that everyone has heard about BPA, that we all have stickers on stuff that says no BPA, you know, go us. But anyway, I'm going to get off my soapbox. Long story short, LH, a hormone from the anterior pituitary gland, is going to trigger essentially this gene to get turned on. You don't have to know this gene, which leads to the production of testosterone. You have to dig pretty, you have to dig for the truth, and you have to, of course, have a pretty strong baseline understanding and understand the science behind things, so you can say, that just doesn't even make sense, or I know that these things here happen, and so it makes perfect logical sense. And also there are these many reputable sources that confirm the fact that chemical A is leading to some funniness with development, with behavior, etc. Yeah, people have a lot to gain from selling stuff with things like BPA in them. Another big one is that microban stuff. Hooray, it kills bacteria, but also it is a very potent thyroid disruptor, right? Like Colgate Total, the total part. Hooray, it's great. It like keeps the bad bacteria in your mouth at bay, but at the same time, again, it's a thyroid disruptor. And lo and behold, we have all of these issues with maintaining weight, being, too heavy or not heavy enough. Go figure, it's all related. Yes, it is very difficult to not become cynical. Yeah, the total part, not normal old Colgate, the total part is antibacterial and also affects the production of thyroid hormones. After grad school, I oh my gosh, I didn't use plastic and I didn't use like everything, but it drives you crazy. It's really hard. You have to pick your battles. And clearly we picked BPA and we got rid of it, hooray. Jury's still out on its replacement. Oh, no. All right. On a similar note here, endocrine disruption aside, as horrifying and fascinating as it is, I want to point out that this metabolic pathway is happening in both males and females. And so, and another thing, testosterone and estrogen are literally one step away from each other. And like, determination of sex and everything is really wishy-washy because we all have the components of both. And especially if you start looking at development, et cetera, could really go either way or somewhere in the middle. And so I want to point out for just a moment that not only is testosterone influencing the development of males and similarly estrogen influencing the development of females, but it's always a bit of both. Certainly, testosterone must be much, much more prevalent in males to get truly male structures and estrogen to get truly female structures. But we all have both. We all have receptors for both, which means that even if you, are genetically male or genetically female, like we are still receptive to both types of hormones. There is no true male hormone or true female hormone. We all need them both. The trick, of course, is to make sure that you have just the right amount of the appropriate hormone at different stages of development and different times and cycles in your life and whatnot, to get optimal functionality as a male or a female. Okay, so once again, testosterone is produced by the Leydig cells or the interstitial endocrine cells in response to LH. LH turns on different genes and testosterone is made. Testosterone does all sorts of different things. First of all, it enhances spermatogenesis, right? So specifically, when ABP binds to the testosterone, right? So essentially the cystinocytes working together with the interstitial endocrine cells, when ABP binds to testosterone, it keeps more of that hormone located within the testis. And the very high concentration of testosterone in the testis is going to stimulate more spermatogenesis. So it's going to ramp up the process and accelerate it a bit. All right, so really you need both LH and FSH to have appropriate sperm count. But FSH is really considered to be the main spermatogenesis hormone, right? So without FSH, you don't have ABP, right? Without ABP, you can't keep all of that testosterone you're making in the testis where it is needed to drive spermatogenesis, right? So if for whatever reason you have low FSH counts, then sperm count might be low as well. Okay. Testosterone, of course, is not the only that does not only act on spermatogenesis, it also has effects all throughout the body. Again, any cell that has a receptor for testosterone is susceptible to its influence. So psychological effects, maintenance of secondary sex characteristics, so a more prominent draw, a beard, different patterns of hair and fat throughout the body. Of course, muscle growth, right? Testosterone generally increases the diameter of your muscle cells, and therefore individuals with more testosterone, that is males, generally have larger muscles than females on average. Testosterone promotes sex organ maturation. And so of course, during puberty, testosterone levels are much higher than before. And so this accelerates the growth of sex organs, both the testes and the penis alike. Again, secondary sex characteristics, erectile function is dependent upon testosterone. Again, estrogen and testosterone are both critical in males and females. It's really just a balance, right? You need good cholesterol, you need bad cholesterol. You can't have just the good, you need both. And similarly, you need both testosterone and estrogen in order to have a functional reproductive system. Behavior and libido as well associated with higher testosterone. And so, as I said, the rising levels of testosterone, of course, enhance all of these different effects, but too much testosterone is also not good. And so the body really tries to hit a target set point. This involves negative feedback as opposed to positive feedback. And so the more testosterone there is, the more there will be a negative influence exerted farther upstream on the anterior pituitary gland. Right, so testosterone is going to put the brakes on this. And so the pituitary produces less of these, which means that now there's less of a signal to produce testosterone. And over a short period of time, less testosterone will be made. Inhibin is something that's produced by the cystinocytes. Right, so these cells produce inhibin to provide kind of a reinforcement of this message. So declining levels of different hormones as you age is just pretty much everything slows down, right? So cell division slows down. So you're not replacing your skin cells as fast. You're not healing as quickly. You're not growing hair as quickly. And so it's the same kind of a thing happening here. Your cells just aren't as active as they used to be. And so there would be less testosterone just because of the natural aging process. This isn't something that happens on purpose. The negative feedback that I'm talking about here is just like from hour to hour for your entire life. You'll want a target set point of testosterone, or at least within a fairly narrow range. And so if the testosterone exceeds that, you want to get it back down to its normal levels. And so individuals who take supplemental steroids, they are essentially adding a ton of testosterone to their body artificially. And so that essentially leads to less of these hormones being produced, right? Because by taking that steroid, you are giving the negative feedback to your hypothalamus and pituitary gland. And so a lot of times these individuals have a lower sperm count because if there's negative feedback up here, there won't be any FSH. Without FSH, there won't be ABP. And so ABP can't keep testosterone in the testis. And so spermatogenesis is going to slow way down. Okay. All right, so GnRH leads to the release of LH from the Leydig cells, which leads to the production of testosterone. GnRH also triggers FSH. FSH talks to the Sertoli cells. The Sertoli cells produce ABP, which binds to testosterone, which together, ABP plus testosterone is going to drive the process of spermatogenesis. Again, just putting a lot of these terms together, the spermatogonium is way up here. One of the daughter cells after mitosis is going to proceed into the adluminal compartment. It is going to then be called a primary spermatocyte. It divides once in meiosis 1. Each one of these daughter cells divide again in meiosis 2. These early spermatids begin the process of spermiogenesis, that is changing shape from these nice little circles here to actually having all the components of a functional sperm. Okay. And finally, the Sertoli cells are going to trim these early spermatids, these spermatids apart and release them into the lumen, surrounded by testicular fluid, again, produced by the Sertoli cells, and therefore washed into the epididymis. Okay, this process takes about 2 months from start to finish, so from stem cell all the way to mature sperm. Let's look at some of the extra components that are added on in the process of spermiogenesis. This part generally takes like 3.5 weeks or so out of that 2 months. So a spermatid is genetically a sperm, but it is not functionally a sperm just yet. And so over the next couple weeks after meiosis is complete, the spermatid is going to lengthen out. So we can see it's no longer round. It lengthens, lengthens, lengthens. And the cell itself is going to differentiate into 3 distinct regions. The first region is called the head. It is triangular in shape and it has a little like drill bit on the top that is called an acrosome. And I say it's a drill bit because this, not only is it shaped, kind of like a drill bit, kind of triangular like this, but also it is what is physically drilling through the multiple layers on the outside of the egg or the secondary oocyte. And so this acrosome or this head is chock full of different enzymes that physically eat through the different protective membranes of the egg. The mid piece, this is essentially the sperm packing itself a lunch. Of course, a flagellum, right, the tail, is very active once it is stimulated to activate, right? Once it's in the right environment, it's out of the male, it's into the female, it is going to swim as fast as it can, as long as it can. And so this activity is very energy intensive. And so the mid piece is essentially chock full of mitochondria. It packs itself a little lunch, so it has a little bit of ATP to get it through. And then once it's in the uterus and in the uterine tubes, the uterus actually feeds the sperm so that they live long enough to get to egg. So again, the mid piece is where the mitochondria are taking sugar, converting into ATP. The ATP is then used to move the flagellum. Okay, these are some healthy parameters like acromial. Oh yeah, I never thought about that before. Yeah, so like the acromion of the shoulder, maybe you could think of, the acrosome being kind of like a shoulder. Yeah, if that works for you, great. However you need to remember, you fit it into your existing schema in your mind. That is good news. Okay, so these are some healthy parameters. Let's see. As Eric was saying, testosterone levels do decrease over the course of your life, but this isn't necessarily on purpose. This is just because everything in the body kind of slows down, unfortunately, but it does. So testosterone levels go down a little bit over the course of your life. Some parameters of semen. So semen being the fluid which contains the sperm, right? So sperm are the cells, the gametes themselves. The semen contains all of the extra stuff, and you're going to learn about that in Professor Fink's lecture as well. Let's see, volume is 1.5 milliliters or more. The pH is slightly alkaline, and a theme that you should kind of get out of this, and I'll bring up a couple more times, is that sperm really likes neutral or alkaline environments, and the female environment is mostly acidic. So for better or worse, acidic female, sperm really like alkaline. And so this is one way of kind of like weeding out the weaker sperm, weeding out, possibilities of making offspring with just anybody. So the female reproductive system is very selective, and one of those ways is to be acidic. Honestly, I don't know why it rises again after 55. I will have to look into that carefully, of course, right? And careful what you Google, especially in this section. Oh my gosh. Okay. One other thing I want to point out, sperm concentration, like absolute minimum, like this is kind of considered to be low sperm count, is 15 million cells per milliliter. So you're talking at least 1.5 milliliters in each ejaculation. And so 15 million per milliliter. And again, that's a little on the low side even. Okay. Okay, so switching to talking about the female reproductive system, since we are getting along here in time. Again, you are going to learn about the external and internal male reproductive organs by Professor Fink, and it sounds like you've already learned about them in the lab as well. Females, of course, also have external genitalia. This is called collectively the vulva. Couple structures on the female. The mons pubis is a fat pad which essentially protects the pubic symphysis and the pubic bones on the female. Finally, sorry. Sorry. The vulva itself also contains 2 pairs of lips. So lips referred to by the labia here. This is Latin for lip. The labia majora or majus is the outer pubic hair covered folds, and it is homologous with the male scrotum. One moment on that. The inner lips of the female vulva are called the labia minus or minora. Within this urogenital triangle, all the way to the anterior is the clitoris. Next is the external urethral orifice, and finally the vaginal orifice. Note that in the male reproductive system, the urinary system and reproductive system kind of share some technology, right? So they share the urethra in particular, but in females, there is no sharing of these different parts. So the urine, the opening to the urethra is anterior to the opening to the vaginal canal. Right. So whatever I don't get through tonight, I will give you guys a recording. Sorry, I know this stuff is really interesting, but this always happens, right? The endocrine system's all the way at the end of A&P 1, 2, and you never really get to talk about it as much as you want. Okay, so homologous. Homologous means structures that have derived from the same kind of primitive tissue, primitive structure. And we could be talking about evolution, or we could be talking about physically within an individual themselves. And so that's where I'm getting at here. In the beginning, we are all the same. So up until we're, give or take 10 weeks post-fertilization, male parts and female parts are the same. And it is the presence of more testosterone or more estrogen, essentially, that is going to drive the development of these same exact structures in one direction or the other. The default is female, by the way. And so the male parts develop essentially from the female parts. And so I find this to be pretty fascinating. This diagram is color coded. So the structures that are derived from the same fetal structures are colored the same. And so here we can see that over time, the scrotum and even the main shaft of the penis is going to be derived from the same exact tissue as the outermost lips, labia majora of the vulva. Within the penis itself is the same exact tissue as the labia minora as well. Also, the glans penis is derived from the same structure, just of course develops to be much larger than the glands of the clitoris. You can't really tell on the models that you guys see in lab, but essentially a clitoris is just a very small penis without a urethra, of course. And so on that note, I know this is wildly off topic from this class, but I gave you guys a couple links here. This sex change operation link is an animation that a scientist made essentially showing how surgically this works. And it's just animated. It's not like real surgery. But I bring that up because it's amazing how in these surgeries, they are utilizing the same tissues, the same technology as our own personal development. So, this, scrotal tissue, right, could be converted into this labial tissue, etc. So very much following the developmental pathway, just kind of in reverse. Also, a lecture on disorders of sexual development. These are much more common than you might expect, or at least I was very surprised to hear how prevalent these are. The fact that that it is so common to have like very male and very female individuals is frankly remarkable. There are so many different variables along the way of your development that can drive any bits of the process in either direction. And so this actually happens quite often. Actually, the most surprising sexual development disorder that I've ever heard of is called testes at 12. Essentially, you grow up female, right? You are physically female until you hit puberty, and then your testes drop. So you are actually genetically male, but that is masked for the first 12 years of your life, and then all of a sudden you become perfectly normal male. Also, there are certain conditions where you can have 3 chromosomes, 2 X's and a Y, and you can express one or the other. So yeah, really wild. That one always stuck with me. But this lecture is kind of cool on that stuff. And lots and lots of information is out there on that. Again, I am amazed that that it is so common to be like so male or so female, and that we're all not just like somewhere in between. Which maybe we are, but that's a different topic. Okay, other female organs. The vaginal canal, also called the birth canal, opens up just posteriorly to the urethral orifice. The vaginal canal is lined with keratinized, stratified squamous epithelial tissue. And so just like every other orifice to the outside world, our oral cavity, our nasal cavity, our anal canal, our urethra, everything is lined with stratified squamous epithelial tissue just because it makes, this tissue is lots of cheaply made cells, no big deal if some slough off with abrasion because of exposure to the outside world, and particularly in this case in sexual intercourse. The hymen is a vascularized membrane present in young girls. And so historically, this was used, or the presence of the hymen was used to determine sexual activity, but certainly this membrane can break in a variety of different ways. On the note of homologous structures, in the male reproductive system, maybe you've already learned this, but there are glands called bulbourethral glands, or Cowper's glands, I think is what they used to be called, that essentially provide lubrication prior to an ejaculation, so at the beginning of sexual arousal, and also is going to increase the pH to make way for sperm, who really like a neutral or alkaline pH. And so in females, those glands instead develop into the greater vestibular glands, also called Bertolin's glands. And these are also going to secrete lubricating fluid into the vaginal canal. And so those would be here. Or just like the colon, the vaginal canal is an ecosystem, right? And in fact, the entire surface of our body is an ecosystem, our oral cavity, et cetera, an ecosystem, lots of different organisms coexisting for the benefit of all. And generally, as long as we are making healthy choices, so that is, we're eating lots of leafy greens, we're feeding lots of fiber to the good bacteria in our gut, et cetera, generally the beneficial microorganisms are going to suppress the pathogenic microorganisms. Now, when this gets out of whack, then you can get different types of infections. And one of the most common in the vaginal ecosystem is a yeast infection. Essentially, this can be caused by a lot of different things, but at this point in life, one of the most common reasons is taking antibiotics. And so if you're taking antibiotics for, whatever reason, oftentimes those are broad spectrum. And so they're not just killing the one thing that you are fighting and really don't want to be sick with anymore. Instead, they are killing off a variety of different bacteria in your body. And so antibiotics might lead to digestive issues, yes, and we kind of alluded to that before, but also in the vaginal ecosystem, there are a lot of lactobacillus bacteria, which, yes, they're also found in yogurts and probiotic supplements and whatnot, but generally you want a lot of lactobacillus. Antibiotics kill off the lactobacillus, and so these bacteria can no longer fight against the most common type of yeast in this ecosystem anyway, which is called Candida albicans, albicans for white, named because of the symptoms of a yeast infection, right? So normally these bacteria are actually secreting a little bit of hydrogen peroxide all the time, just as a byproduct of the things that they do. Lactobacillus is fine with hydrogen peroxide. Candida albicans is not. And so if you throw off this ecosystem in any way, and not just because of antibiotics, but that is very common source at this point, then other organisms can increase in prevalence. Male prostate to the female. That is a great question. To my knowledge, there is not a homologous structure to the male prostate in the female. So many good questions tonight. I'm gonna write some of these down because I am honestly not sure. Let's see. Yeah. So that's another good question. So when sperm enter the vagina, they release something to slow the immune system. That is also true. Semen has so many different ingredients that promote, sperm's life and contraction of the uterus to essentially push the sperm up towards the fallopian tubes. Semen contains relaxin, right, to actually relax the entire female pelvis and therefore open up the female reproductive system for the entry of sperm, right? So semen has all sorts of different things that's really fighting for the sperm, and then on the other hand, the female reproductive system is fighting, like her immune system is like, no, you can't come in. These are foreign cells. Oh no, you know, the acidic environment, etc. And so one of the components of semen is to suppress the female's immune system so that her white blood cells don't just munch up all of this, all of the sperm before they can finally get to their destination. Okay. Great questions tonight, everyone. Good stuff. Anything else while we're here? All right. Just writing down some of these questions so that I can do some careful research. All right. So final detail here about the vaginal canal is that it functions both in copulation, so in sexual intercourse, as well as in childbirth. And so it has a lot of flexible functions for those purposes. The uterus is a hollow, thick-walled muscular organ. It is anteverted. And so I feel like in high school sex ed classes and, you know, even at the gynecologist's office, you know, images online, whatever, you generally see the uterus portrayed like this. So nice and up straight and uterine tubes way out to the side. But in reality, here is the uterus, which sits directly on top of the urinary bladder. So you can imagine that if there was a growing fetus inside the uterus, no wonder mom has to pee all the time, right? There's just not as much space. Furthermore, I don't know if you've noticed by looking at the models in the lab, but females have a whole extra set of organs here. Males can use that extra space for urinary bladder space. And so she has to pee on a road trip. Like, it's really not her fault. It is a smaller bladder. All right, just throwing it out there. Okay, so the uterus itself is, yes, anteverted, which means it's flopped down on the top, on top of the urinary bladder. There is a main body, and so this is where implantation of a fertilized egg would ideally occur, and also where the development of the fetus and the placenta would be. And just like we saw in the stomach, the uterus is kind of domed, right? So that is called the fundus. The opening from the vaginal canal into the uterus is called the cervix. So yes, we, you know, or in AP1, you talk about the cervical region, the neck region, the cervical spine. So cervix just means the neck. So the cervix, right, the neck between the uterus and the vaginal canal is lined by glands. And so this is yet another way that the female reproductive system kind of changes based on, well, you take a step back. The female reproductive system essentially blocks the entry of sperm into the uterus when she is not fertile. Right? Essentially, the entry of semen into the uterus pretty much means that the female's immune system has to clean up all of those sperm. It has to clean up all of the other ingredients of semen. And so if she is not fertile, she doesn't want to have to clean all that up. And so the cervical glands actually secrete mucus that is fairly thick, and it very much blocks the penetration of sperm, except for the couple days a month when she is actually fertile. So block sperm entry until mid-cycle, and hopefully we'll get into that tonight or minimally on Thursday's lesson. Okay. Again, a theme that we have seen literally from the first week of class together. The wall of this organ is composed of 3 different layers, right? And the tissue matches the function of each of these layers. All the way on the outside is the perimetrium. Right, so peri going around the perimeter. The uterus is retroperitoneal. So the peritoneum kind of sits on top of the uterus, but it's not surrounding the uterus. Okay. The thickest layer of the uterus is called the myometrium. So myo meaning muscle, right? So this is very, very thick, smooth muscle. Of course, for the shedding of the endometrial wall during a menstrual cycle, and also for contractions and first of all, eviction, the release of a fetus, of an infant. Finally, the endometrium, endo meaning all the way on the inside. This is a mucosal lining. This, or at least part of this, is what sheds during menstruation. If there is a fertilized egg, that will implant in the endometrium. So it physically grows into this layer of the uterus, and it forms its own blood supply within the uterine wall. Yeah, I did say shed. I think that's what your textbook says. And it is shedding, actually. And yes, that would be interesting if this was more of an equal opportunity experience, but we all know pros and cons to both, I think. Okay. The primary sex organs of the male are the testes, right? Testis for singular, testes for plural. I'm sure that'll come up on your practical exam, which is coming up really soon here. The primary sex organs of the female are ovaries. They're just a few centimeters big, so maybe smaller than you might expect. They have three main functions, just like in the testes. They produce the gametes. The gametes themselves are called oocytes, right? And that's honestly how I've heard it pronounced, like, you know, all through school. It's not oocytes, it's oocytes. Yeah, seriously, guys. Okay, so the process of generating oocytes is oogenesis, right? So spermatogenesis, generating sperm, oogenesis, generating oocytes. Furthermore, the process of oogenesis in females is, as you might imagine, substantially more complicated than developing sperm in the male. It's not just the process of meiosis that happens over a few weeks' time. Instead, this process actually begins before you're even born. All right, we'll get into that here in just a moment. But also, each and every one of these cells is surrounded by something called a follicle. All right, these follicles, if we look in the ovary over here, these follicles are a series of cells that are nurturing, that are making all sorts of hormones, that are essentially regulating the entire process and can actually sustain a pregnancy if that happens to occur. So I feel like we all learn about sperm and egg, but very rarely in like your normal high school sex ed do you ever learn about follicles. So this is something really important to concentrate on. Right. The ovaries are responsible for producing estrogens and progesterone. Remember that all of these hormones are very closely related. They are like a single step away from each other. So estrogens and progesterone are the main hormones that are going to play a role in regulating female cyclicity, behavior, development, etc. Just like in males, the gonad of the female produces inhibin. And so inhibin is just one more hormone that can kind of put the brakes on the brain and the pituitary gland to kind of stop the signal to produce more hormones. So oogenesis. This process, unlike in males, begins before she is even born. So literally, the half of you that came from your mom, right, the egg that made you eventually, was literally started, right? It formed in your grandmother. And so I'll say it again, you started your formation in your grandma, which is kind of crazy, but that's how that works. Yeah, right? Cool stuff. And so this process starts before birth, and then it stops, and it's paused for years. And then every month from puberty until the time you hit menopause, in theory, a single one of these eggs is selected through some mysterious process to this day we don't understand, but some of these eggs are selected and they continue the process of meiosis. So it begins in the fetal period. And so we don't have to have a barrier between the soon-to-be gametes and the rest of our cells, because our immune system develops being able to look at these, right? So they have started well before. As long as you can go before hitting menopause, that is also a great question. I'm not sure. It really varies according to genetics and lifestyle. And I'm sure that there are supplements that you can take to extend your menstruation time. I'm honestly not sure when like generally the cutoff is. And even if there was like a specific cutoff date, I'm sure there would be outliers as well. Okay, so the process of oogenesis begins with stem cells, again, just like we saw in the males with spermatogenesis. These stem cells are called oogonia. All right, so 2N, they are the diploid ones. They are genetically identical to every other cell that's located within your body. As a fetus, right, in female ovaries, essentially these oogonia undergo mitosis. Some of those daughter cells are going to develop into primary oocytes, and the primary oocytes begin the process of meiosis 1, specifically prophase 1. So they literally start the process, all of the chromosomes line up, and that's it. Okay. And so these cells are then arrested in this particular phase, in literally the very first step, until puberty. Or if we're talking about individual cells, so one individual cell is arrested until, you know, October, when woman is 39 years old, right? So sometimes these eggs are arrested in this phase for many, many decades, right? And so if menopause doesn't happen until maybe 50, 55, 60 years old, these eggs have been arrested for 60 years in one place. And so that's why it increases the risk of, you know, having offspring with abnormalities, right? So genetic mutations, and also too many chromosomes are not enough. So Down syndrome, when you have older women reproducing, just because these eggs, they've been sitting there literally arrested in place for many decades. And so in addition to beginning the process of meiosis, follicular development begins as well. And so primordial follicles are essentially one skinny little layer of cuboidal cells that surround the developing egg. Not really doing too much, they're just kind of hanging around with a shell around the oocyte. When girl is born, she has a lifetime supply of her oocytes. No more are going to be made over the course of her life. What you're born with is all you ever get. And so generally, girls are born with about 2 million of these eggs, or primary oocytes surrounded by primordial follicles. Remember that with every milliliter of semen, there's about 15 million, right, of those cells. Girls are born with 2 million tops for your whole life. And by the time you hit puberty, you only have about 400,000 left, because they do die off over time. But keep in mind, like, even if you do the math, you know, you are menstruating for so many decades, one menstruation a month, give or take. And so generally you only need to ovulate about 300 eggs. It just seems like it is so very limited compared to spermatogenesis. Okay, so again, every month after puberty, a few of these oocytes are activated. Again, we have no idea why certain ones are activated and other ones aren't. Of those few, one single egg, in theory, unless you're on some kind of fertility treatment or something, is selected each month to resume the process of meiosis. So here is the primary oocyte. It has been frozen in place for decades, maybe. And only after it is selected can it continue the process of meiosis 1. Okay. Unlike in spermatogenesis, after meiosis one, what is produced is two unequal cells. One cell actually contains the cytoplasm and all of the nutrients, all the goodies from both of these cells. There is a second smaller cell, which is pretty much just there to hold on to the extra DNA, right? The goal here is to have haploid cells. And so this first polar body is just taking the chromosomes out of the equation, out of this cell, and donating everything else that is in the cell, all of the organelles, all of the nutrients, all the contents. Okay. So a smaller cell, this secondary oocyte, right, which is also going to be arrested. So again, all of the chromosomes line up for a second time for the second half of meiosis, and then it stops again. And so this is actually what is going to be ovulated. It is arrested in metaphase 2. And in fact, if a sperm doesn't fertilize the egg, meiosis 2 is never completed. So you never actually get the final division of all of these different cells, 3 super tiny cells, 1 really big ovum egg cell. The only way that happens is if fertilization occurs. And I am out of time for this evening.